No, thanks. Good morning. I think we'll start. Like you can go out and have an earlier lunch, right? How's everyone? Everyone okay? It's Friday morning, right? A week almost done. So this lecture, you know most of the things I'm going to talk about because we already talked about them during the immunology. And again, here I won't be going very deep into the detailed mechanism. What I would like to do is actually put in perspective the different part of the immune system you've learned about and how they can interact with how they used to fight bacterial infections, okay? And I divided in two parts, actually 3 parts. The first one is what are the important mechanisms to fight extracellular bacteria, okay? Second part is what are the important mechanisms to fight intracellular bacteria. And in the third part, I will just give you example of how bacteria basically can escape those immune strategies, okay? So I would not ask you to remember the different toxin or genes that are used by bacteria to evade the mechanism. What's important is trying to keep in mind when there's an extracellular bacteria, this happens most of the time, okay? The general ideas. And before we talk about the different immune response to extracellular and intracellular bacteria, just a brief description. Those you know much better than I do, the different routes of infection and the different steps of bacterial infection. So you know we can get infected through the respiratory tract, through the gastrointestinal tract, through the genitourinary tract, and obviously by following any injury, any unnatural routes, okay, that a wound that's open, bacteria can basically cross the first barrier we already described in 331, like the epithelium, right? Everything that enables bacteria to reach inside the tissue is a good way for the bacteria to invade. So those are very general. But when we think about the bacterial infection, we have to consider there's different steps in that. The first thing is the colonization, okay? Basically, bacteria needs to attach to the substrate, being the epithelium on your skin or in your GI. They need to attach somewhere, and they will usually, the attachment will enable them to persist in the environment. Otherwise, they can be just washed out, right? If they don't attach, you move them away. So they have to persist and then grow, okay? Then you have colonization. If it stays on the epithelium, it's usually not a problem, but a pathogen, especially professional pathogen, I've evolved ways to basically invade. So the second step, so getting deeper into tissue, okay? Getting nutrient deeper into host tissues. And so that's the second step, the invasion. And this invasion will basically result in damage, right? Tissue injuries. Because of those tissue injuries, due to the bacterial growth or because of the bacteria will produce toxins, okay? The presence of the bacteria itself, you know, will produce PAMs. You will have cells that will produce pro-inflammatory cytokines. The bacteria themselves produce toxins. So all that will trigger inflammation, okay? And this inflammation, again, as I just said, can be initiated by the complement, by damages, toxin, the bacterial growth, or by antibody that will bind to the bacterium. So that's the first 3 major steps of bacterial infection. Attachment, or colonization, invasion, and then generally you have inflammation. When you see pus, and then you will have recruitment of all those inflammatory cells. What's important to keep in mind is that the damage associated with inflammation are not always due to the bacteria. Okay, the bacteria will produce toxin that can damage your cells and your tissue, but the inflammation itself, the immune cells, will also be responsible for tissue damage. Okay, so remember briefly the M1 macrophages and the M2. The M1 would be responsible for killing, and when you try to kill bacteria, you will produce molecules that can also kill your own cells, and then you would need the M2 to fix it later on. Okay, so damages in general are not only produced due to the bacteria. Okay, and it's true not only for bacterial infection. Actually, in a lot of situations, when we get very sick, it's also because our immune system responds too strongly to a microbe. Okay, so those are very general information, but let's look at what are the important immune mechanism in response to extracellular bacteria. So when I'm talking about extracellular bacteria, is bacteria that will not have evolved to persist and survive within cell, okay? Something that can grow outside. Like you think E. coli, Staph aureus. Even though Staph aureus can do both, so if you remember my cartoons from the immune lectures, one of the first things that after passing the epithelium, going through the mucin and the defensin, one of the first mechanisms that will be triggered in response to microbes is basically the complement activation. So remember, there's two ways that a complement can be inactivated in the innate immune response would be through the alternative pathway, C3b, C3b, BB, C5, okay? But you know that already. And the lectin pathway, where you will have the mannose binding lectin here that recognize mannose on the surface of bacteria, or LPS, or peptidoglycan, and the classical pathway. In this case, if we talk about the innate complement activation, those antibodies will be called natural antibodies. I think by now you have heard about that, right? Natural antibodies? Okay. So I think in the handout you might have C2A here, but remember what I put on Facebook, C2A, C2B used to be called C2A, so it's fine. Then you have, production of the anaphylatoxins, and basically the consequence, as you remember, of the bacterial activation is the formation of the membrane attack complex here and the lysis of the bacteria. But the activation of the complement also leads to the opsonization of the bacteria, right? The C3b you find on the surface of bacteria is an opsonin that will enable the interaction of the more efficient recognition of the bacteria by the phagocyte. And again, the C3a and the C5a will be potent molecule to recruit more leukocyte, okay? So macrophages, monocytes, or granulocytes will be attracted to the site of invasion. So nothing new so far, right? Everyone okay? Now, we talked about some of those receptor during the immune response, innate immune response lecture I gave. But actually we can, here I'll try to show you that not all receptor on the innate cells behave in the same way. There's some receptor, such as TLR, that's really involved in signaling and activating the cells. So the consequence of the TLR or the node-like receptor, as you remember, and all those pattern recognition receptor, is mostly to stimulate the bactericidal activity, which means that those macrophages will start to express, for example, the nitric oxide synthetase, the NOS, okay? Or they will start to produce more reactive oxygen species, okay? Some other receptor will be playing a role mostly in phagocytosis, so internalizing the microbe. That would be the case of the mannose receptor we also described, and the scavenger receptor. Those are found on the surface, and when you have phagocytosis, through those receptors, usually the macrophages or the neutrophils will not get activated in a bactericidal way, okay? But there's some receptor that actually, those receptors will just basically stimulate phagocytosis and enable the internalization of the bacteria. So again, if bacteria is recognized by a TLR, usually it changes the macrophages, it activates him, okay? But TLR is not involved in phagocytosis itself, right? If phagocytosis is performed by mannose receptor or scavenger receptor, but those receptors will not activate the macrophage. And there's some receptors that are important. They recognize opsonin. Those receptors will be able to both stimulate the phagocytosis and the bactericidal activity. So that would be the case of CR here, which is a complement receptor, and the FC receptor, or immunoglobulin receptor. So in a way, it makes sense, right? Those opsonin receptor recognize opsonin. So if you have opsonin, it's on the surface of a microbe. If you have antibody that recognize a microbe, it's mostly a pathogen. So you have to activate the bactericidal function and phagocytosis at the same time. So antibodies and C3b, for example. There's something I haven't described in the previous course. It's called neutrophil extracellular traps. It's something that's been discovered about 10, 12 years ago. People are not, there's still debate about the real function of those neutrophil extracellular traps, but I'll talk about it because they're also associated with a lot of pathologies, okay? So when you think about neutrophils, again, as phagocyte, we know their principal function will be to phagocytose bacteria, to kill them, or to degranulate, which means they will release the bactericidal content outside to kill the bacteria that are outside. There's another mechanism in which they will do this weird thing, which consists of mixing their DNA, here in purple, with the content of the granules. They will mix that, and they will release it, okay? And because you know that the DNA, when it's not packed into chromatin, it's very long, right? Very big. So when it's in this form, the DNA covered with the content of the granule, it can cover a larger surface than a neutrophil itself. So it's thought to be involved in preventing the spreading of bacteria, trapping them in a net, if you want. And so that's why they call them extracellular traps. And in some cases, kill the bacteria, because you have the same content that you found on the granules, you will find it on those nets, okay? So as you see here, nets are composed of the DNA plus the bactericidal proteins you find in the granule. and everything is mixed within the cell, and the cell bursts out and release those. I just have a video just to illustrate that. There's no bacteria here. What you'll see is just the DNA. The DNA will be staining green, but what you will see is only the DNA that will be released. If the cell is intact, the dye cannot stain DNA, okay? And this is not in response to bacteria, it's response to a drug that's known to induce NECs, but that gives you an idea of what can happen. So initially you see very few cells are green, and over time some start to pop. And you see that this DNA spread on very large areas, right? So I don't know if you want me to play it once more? Or okay. So that's those mechanisms. Another way the innate immune system can fight the bacteria. And this one I probably go quickly because you certainly remember everything about it. The different part of the phagosome where the bacteria is eliminated within the phagocytes, you will find, again, the NADPH oxidase to produce high amount of reacting oxygen species, hypochlorite acid right here. You will have formation of inos, okay. You will have a starvation of the bacteria through chelation, for example, of iron through lactoferrin. And you have a lot of proteases and peptide that will damage the membrane of the bacteria and compound that will damage the DNA, okay? So that's mostly those mechanisms we already covered, but those are important weapons in destroying the extracellular bacteria once it's been internalized. Now, when we think about the humoral immunity or the adaptive immunity, humoral response to extracellular bacteria, obviously we'll talk about antibodies. Okay, so how do antibodies fit in this story? Mostly, if we look at extracellular bacteria and the toxin it can produce, so toxin and can be either secreted or found on the cell membrane. Antibodies can do many things. First is would be neutralize the bacteria or the toxin. Okay, when we say neutralize, we mean that it will prevent the colonization, prevent the interaction of the bacteria with cells. Okay, so the bacteria cannot just attach itself and cannot grow. Okay, cannot persist. It can also, just by binding to the toxin, it can inhibit their function, right? So neutralizing the bacteria and the toxin. Those antibodies can also obviously perform opsonization, right? They bind on an antigen on the surface of the bacteria, and on the other end, they will bind on the Fc receptor on the surface of a phagocyte. Will it be a macrophages or neutrophil? It doesn't matter. You have opsonization, eventually leading to Fc-mediated phagocytosis, right? And eventually elimination. A third major function of those antibodies is, again, activation of the complement. This way, we're not talking about the innate activation of the complement, we're talking about the classical pathway, again, through C1q, and as you remember, the cascade then is very similar to the lectin pathway. And this by itself will obviously also lead to opsonization because you will have activation of the complement. Inflammation, you will produce anaphylotoxins, and eventually the lysis of the bacteria, okay? So again, nothing new here, but just putting in perspective. Everything okay so far? Yes? All right. let's move on then. We'll talk a little bit about the T-cell mediated response. So you remember your lecture about antigen presentation and T-cell activation, right? So this is very simple, highly simplified, but if you have a dendritic cell that capture a bacteria, a microbe, basically remember that it gets activated and moves to afferent lymph node and then start to present antigen where it can potentially meet a naive T cell, okay? A naive T cell move from lymph node to lymph node until it find a match. When that happens, this T cell can differentiate into different type of T cells, the helper. After proliferating and differentiating, it can become the follicular helper T cell. As you remember, it's the one that helps B cells to produce high affinity antibodies and do the isotype switching. But you can also have different subtype of helper T cells. As you probably heard about the Th1 subtype, the Th2, the Th17, and there are more Th9 and so on. But I'll just talk about Th1 and Th17 here. Why? Because as you remember, Th1 are a major producer of interferon gamma, right? So when you have a Th1 T cell that's interacting with the macrophages that has already phagocytosed the bacteria, but not been able to eliminate the bacteria, the macrophage as an antigen-presenting cell represent the peptide for this helper T cell that's already been activated. It's not naive anymore, it's effector, right? So production of interferon gamma and eventually activation of the macrophages, and you will have the destruction of those bacteria in most cases, okay? So that's how helper T cells, especially Th1, will be important for the elimination of bacteria in general, but especially extracellular bacteria. Th17 will also play an important role in that by producing the IL-17 cytokines that can lead to the recruitment of granulocytes, and mostly neutrophils, okay? So that would lead to inflammation, again, recruitment of neutrophils, and very importantly, induction of the granulopoiesis, which means that the bone marrow will be producing high amounts of granulocyte. Okay, so you will be producing more neutrophils if you want, in case of infection. So providing more weapons to fight the bacterial infection. Now again, those cell type don't, their function is not limited to extracellular bacterial infection, okay? But that's how they would play a role in bacterial infection. Okay? Now let's talk a bit about the immune response to the intracellular bacteria. And again, when I talk about intracellular bacteria, I don't talk about the one that are phagocytosed and easily eliminated. I'm talking about the microbes that have evolved to survive inside cells, okay? So some of them can survive in the cytoplasm, escape from phagosome or endosome, and survive in the cytoplasm of mammalian cells. Some of them can even survive in the vesicle or even in the phagolysosome, okay? So that's what I'm talking about. And here you have the major component we'll be talking about. So the NK cell, the macrophages, the cytokines, interferon gamma, IL-12, IL-15, a ligand for NK cell, MYC-A, and this is a horse. You probably know why already, but if not, by the end of this lecture, I'll say why I picture a horse here. So, as we discussed, most of the innate immune cells, the phagocytes, express a lot of pattern recognition receptors. Okay, the node-like receptor, the toll-like receptor, that will basically activate them and recognize they get activated whether you have a microbe inside vesicles, like here, or inside the cytoplasm. Okay, you have the node-like receptor, as you remember, the NLR. So without going back to the signaling pathway we described in 331, but you remember it, so it's fine. Those phagocytes will basically will get activated, okay? And the same weapons I described just before, the one you find in the phagosome, will be used to destroy bacteria if they reside in phagolysosome. If they are in the cytosol, there are actually other mechanisms involved. I actually realize I don't have the slide about it, but if you're curious, I can explain later. If the bacteria infect normal cells, non-phagocytes, then the principal axis that will be involved is a macrophage NK cell axis when we think about the innate immune response, okay? So the intracellular bacteria, just like viruses, just like every pathogen, will trigger the expression of stress molecules, right? Including, for example, MIC-A on the surface of the cell. And as you recall, MIC-A is one of those molecules that can activate NK cells, right? And intracellular bacteria can also induce the expression of IL-12 and IL-15 in macrophages and dendritic cells. So those cells, when the toll-like receptor are activated, when the node-like receptor are activated, will produce those IL-12. And as you remember, that will lead to the activation of the NK cells. So together with the interaction with MIC-A, you will have a very potent cell that will essentially kill the infected cell, right? So as you remember, the NK cell will induce apoptosis, and so the bacteria that are hiding within the cell, basically they don't have a house anymore, right? The cell is dying, so there's no nutrient, nothing. The other consequence of the activation of the NK cell is, again, the production of interferon gamma that in turn will trigger the activation of the macrophage. So we're always talking about the same cytokines here. IL-12, interferon, and making those macrophages more potent in their bactericidal activity, okay? So basically, again, when you have bacteria within the cytosol of cells, they either express MIC-A, or if you talk about immune cells, they will produce pro-inflammatory cytokines that will activate NK cells, and there's a positive feedback loop that can basically activate macrophages in return. Another aspect is to look at the adaptive immune response to intracellular bacteria. And that will mostly be T-cell mediated, right? You have a bacteria that's inside a cell. So antibodies are basically useless, right? Antibody cannot reach the bacteria inside a cell. It can be effective only once the bacteria is outside. So how do we get those bacteria from inside to outside? That's the question. So we just, on the previous slide, we already described that. Intracellular bacteria will induce the production of IL-12. And that IL-12 not only activate NK cells, but also can lead to the differentiation of T cell, CD4 T cell to Th1 subtype, okay? So cytokine together with the recognition of a peptide on MHC class 2 will promote the Th1 differentiation of the T cells, okay? And we already covered that a number of times, but those Th1 cells, the major cytokine they will be producing is interferon. So you have the same mechanism here again. IL-12, interferon, activation of the macrophage, right? And in most cases, that would be enough for the macrophage to eliminate intracellular bacteria. But when we're talking about those professional intracellular pathogen, some bacteria might persist. So there another facet to this adaptive immune response to intracellular bacteria, that would be the CD8. What role can they play here? So what happens if the bacteria survive in the cytoplasm? Remember that macrophage, just like any cells, any nucleated cell, also express MHC class 1, right? And MHC class 1, as you recall, is recognized by CD8 T cells. Now, if the CD8 T cell has been, is an effector cell, so already been activated by dendritic cell in a lymph node, it will recognize this peptide presented by MHC class 1. And eventually, what is the function of a CD8 T cell? It's just like an NK cell is to kill abnormal cell. In this case, basically, it will induce the death of the macrophage or any other cell types. And again, when you induce the death of the host cell, the bacteria is exposed, right? It will not be found within the cell anymore. It will be exposed in the extracellular compartment where you have complement antibodies, okay, that can eliminate them. So same thing will happen with NK cells. When they lyse infected cell, the bacteria might not die because of that, but they will be exposed to other parts of the immune system that can eliminate them, okay? And another consequence of the activation of the CD4 T cell is basically the humoral immune response, right? Those cells that have been activated by IL-12 will, this is extremely simplified, right? But eventually they will produce a cytokine that will activate B cell, trigger the class switching, okay, and the memory plasma cells. You will have production of high affinity antibodies, and that will play a role back toward those bacteria once they're exposed, okay? Okay? Any question? No. So again, this is extremely simplified, but it's 1 hour, okay? So, as usual, if everything was working, if the bacteria were not striking back, we would not be here talking. We wouldn't need doctor most of the time, right? But obviously, as the system, immune system evolves to eradicate microbes, microbes evolve to survive. Okay, so that's what I try to illustrate here when basically the bacteria strike back. And what I do, I'll just again give you examples. I don't want you to remember, I don't need you to remember those different factors, but it's just to illustrate that, again, everything I describe as very effective, very potent, there's at least one microbe that find a way to inhibit or prevent that to happen, okay? So if we start at the beginning, like the best, probably the strongest and best defense against bacteria once they invade the complement, you find it everywhere. Well, if you just think about, for example, one you're very familiar with because you've stained it 2 or 3 times already, in the lab, right? SREs. This little bug is able to remove the C3b you find on its surface. So in my immunology lecture, I lied a bit. I told you mammalian cell can remove C3b and most of the microbe cannot. Well, most of the non-pathogenic microbe cannot, but the pathogenic one can. Okay, they usually have ways to remove C3b, and the name of this protein would be the staphylokinase, for example. That just removes C3b, so you will not have formation of the alternative pathway complex, C3b, BB, okay? Now, you might wonder, well, SREs can prevent the alternative pathway, but we still have the mannose pathway, right? Well, or even the classical pathway that use C4b, C2b, C3b. Well, Staphylococcus aureus produce a protein called SCIN that can prevent the formation of this convertase. So basically, no alternative pathway and no classical or lectin pathway. Not only because you block the formation of convertase, but in the case of SREs, it can even prevent the interaction between antibodies and C1q. Remember the big complex that initiate the classical pathway. This interaction is inhibited by another factor of SREs. We're still talking only about SREs, okay? But another one is, obviously they won't be able to block everything at once all the time, otherwise we'll be all dead, right? But you have overlapping mechanism like this. One other is the ability to antagonize C5a. So if the complement is activated and you produce the anaphylatoxins, C3a, C5a, SREs can produce a protein called CHIPS that will bind to the C5a receptor and prevent C5a to interact. So you will not have recruitment of the phagocyte to cytome infection. A few more examples. We're thinking about the MAC. Streptococci can actually prevent the MAC formation. They express a protein called SIC that will prevent that. So again, we look at every single part of the complement pathway. Again, there's one bacteria which one way or another can prevent that. The MAC, the C1q, the mannose, and classical pathway, the alternative pathway, the production of the anaphylatoxins. I also just mentioned in the previous slide the formation of those extracellular traps, right? Those funky, weird structures that are composed of DNA and bactericidal component. Well, there's many bacteria. Since their discovery, people have shown that a lot of pathogenic bacteria secrete DNases or express DNases on their surface. And what that means, for example, in the case of Vibrio cholerae or in case of Streptococcus, you have DNase, XDS, and SDA1. But those are just a few examples again. Once, again, SREs, it's everywhere. Those DNAs, what will they do? They will basically degrade the backbone of those nets. They will remove the DNA. Okay. And then you have bacteria that are more or less free now. Okay. Instead of being trapped, they can use the DNA as blocks to grow, and they're not trapped anymore. Okay. They can disseminate. When we think about the humoral immunity, one of the major ways those bacteria can evade it is through antigenic variation. So maybe the most infamous bacteria in this genre will be the Streptococcus. where you have always a plethora of serotype circulating at the same time. When if you get infected with one serotype, so your innate immune system try to eradicate or slow down the infection, while the adaptive immune system takes its time and produce very good antibodies to basically eliminate this bacteria. But in the meantime, the bacteria may be able to infect other strains. And your very clever adaptive immune system produce memory cells, but the bacteria, they're a big family. And next time you're exposed to a new serotype, it's useless, right? So leaving enough time for this bacteria to colonize, infect, you eradicate it, but it has probably enough time to infect someone else on the way. And then you have antibodies, and you have memory against number 2. And if you're not lucky, you might go on for 3, 4. Okay, that's unlikely, but. So every single injection, when you face a microbe that has a lot of antigenic variation, you require a new primary immune response and secondary. You need to create new antibodies every time because the immunogenic and the major immunogenic antigen are not the same. Among those bacteria, you have obviously Streptococcus pneumoniae, E. coli, Neisseria, and Salmonella are famous for those antigenic shift or variation. Now I talk a lot about SREs, but this is a model that, it's mostly because it's a model that's used a lot in the field. It's easy to grow, it's easy to study, so that's why people probably understand a lot of things about SREs, but what I describe is probably true for a lot of the pathogens, okay? I slightly modified the slide, but again, don't worry too much about the genes or the molecules I'm talking about, just to illustrate it. So, as we discussed, immune cell will have those activating receptor, the toll-like receptor, for example. That's once it binds to a pump, will activate the macrophage. But they also have inhibitory receptor because you need a way to control the innate system, immune system, even though I did not describe them much, or not at all actually. But some bacteria take advantage of that. So as you remember, the pumps, they are essential, right? Bacteria cannot grow without them. It's an essential molecule for bacteria, so they cannot get rid of them. They will always have them. It's essential. So what some bugs have evolved to do is they still express a pump, but they will also express a mimetic that will bind to an inhibitory receptor at the same time. So you will have the pump that will activate the toll-like receptor, but the same bacteria will produce a molecule that will bind to an inhibitory receptor at the same time. And essentially, plus, minus, 0, right? So the overall effect of that is you don't have inflammatory response, or it's inhibited. So in this case, one example would be the lipoic acid expressed by SREs. The receptor will be TLR2 and PIRB, okay? This is another example. This is the same concept, except that those two receptors are not, one is not an inhibitor and the other one is activator. They are two receptors that cooperate, okay? So when both receptors are interacting, the activation of the cell is higher. In this case, you will have the Fc gamma receptor, the complement receptor can form a complex in response to binding to their own ligand. And bacteria such as Streptococcus group A can produce, again, a decoy or mimetic that will prevent the interaction between those two receptors. So the consequence is the same. You will have maybe activation of both receptor, but the activation when they're isolated is not, the effect is not as strong as if they are cooperating, okay? And in this case, if you prevent interaction between the complement receptor and the Fc gamma receptor, you decrease or inhibit phagocytosis, which is good for the microbe, right? You have, so we already described how bacteria can alter the complement activation. So that falls also into the inhibition of phagocytosis. As you remember, the complement produce opsonins, right, P3B. So without activation of the complement, you won't have phagocytosis. And that includes the streptococcus and the SRAs already talked about earlier. Another way to evade phagocytosis is basically to be encapsulated. So have such a thick layer of lipids or sugar on the surface that prevent basically the interaction with opsonin or with mannose receptors or things like that on the surface of the phagocyte. The example of bacteria that do that are the streptococci and Neisseria. Basically, so thick around them that the receptor on the surface of phagocytes cannot even reach the ligand, okay? And this slide, you're very familiar with it now, at least the left side. You've seen it at the third time I use it. But everything I've shown in this part, the NADPH oxidase, the lactoferrin, the peptides, obviously there's some bacteria that have evolved a way to block it. For example, if you look at the formation of the NADPH oxidase complex, you might not remember, but you have phosphorylation of P40 and formation of the big complex and all those things. Well, the Coxiella can basically inhibit it, okay? Those are antibacterial peptide that loves to bind to the surface of the bacteria because of the charge. Well, SRAs express a protein that's positively charged. So those peptides, which are also positively charged, will be repulsed, removed. Even though you're able to produce H2O2 through the NADPH oxidase, you remember you did that Monday, I think. The catalase assay, the oxidase assay. Did you have to do the catalase assay, right? So now you know why. Why do they express this catalase? Well, it's basically to degrade the H2O2 produced in the phagosome of phagocytes, okay? So SRAs produce catalase to get rid of this H2O2. And some bacteria that are very adept at surviving within phagocytes, such as Mycobacterium, they can even block the formation of inos or block the recruitment of inos to the membrane of the phagosome. Okay, so you won't have nitric oxide produced. Okay, this is very toxic, and so they just block the production of inos, prevent it. So again, everything you see on this side, there's a way for pathogens, so the bacteria that have really evolved to fight our immune system, to hijack them, all those mechanisms. And actually, most of those microbes, so you have the microbes that try to evade phagocytosis, but you also have the one that want to be phagocytosed, okay? And so there's a way for some of the bacteria to manipulate the immune system, okay? Or cells in general. So if we look at the regular, the expected result of phagocytosis, right, is formation of phagosome, then formation of the phagolysosome, and eventually the degradation and destruction of the bacteria, right, in the professional phagocyte. And as I just mentioned, most of the pathogen we're dealing with, they're actually fine with being phagocytosed. If we talk about Salmonella, it will induce itself phagocytosis. The interaction of some protein on its surface will lead to the formation of a phagocytic cup, and it will be internalized into the vacuole. Listeria, even though it's not on the surface of immune cells most of the time, it will bind to a specific receptor on the surface of cells to be internalized. So the reason why, it's because they can survive inside, right? Some of them will prevent the maturation of this phagolysosome, of this phagosome. So it will not fuse with the lysosome. That's the case of Salmonella or Legionella. They will get phagocytosed, but when it comes to the fusion between the phagosome and the lysosome, that will be inhibited. Those proteins will those bacteria will secrete proteins that will block the signaling pathway of the host cell. You have some bacteria that actually can escape from the phagosome. That's the case of Listeria. So you might have heard of Listeria lysine, right? That basically it's a molecule, a protein that makes pores in the membrane. This Listeria lysine helps the Listeria to get out of phagolysosome, and end up into the cytoplasm of cells, which is not as harsh environment as the phagosome, right? And some do that because now that they're inside, they can stay inside and can infect, move from cell to cell this way. That's the case of Listeria, that's the case of Burkholderia, Pseudomallei you've heard, that can use the same approach. When they are in the cytosol, they basically can move from cell to cell and spread like that. And then you have the king of the hill, the one that bring me to the phagolysosome, I don't care. That would be the Mycobacterium, okay? Survive for a very long time within the macrophages. I'm fine, I'm chilling. I'm not growing fast, but I'm okay, okay? That would be the case of Mycobacterium. That's just a few examples that even this complex system, the way we're trying to destroy, the immune phagocytes are trying to destroy those bacteria. There's, again, always a counterexample of bacteria that can do the, evade that. So the question is, why would they want to get phagocytosed in the first place? Why hijack the phagocytes? So the first one is obvious. You have protection. You're protected against antibodies. When you're inside the cells, there's no recognition by antibodies, so you cannot be neutralized, okay? Your toxin cannot be neutralized. The complement cannot reach you. Most of the time, those bacteria will survive in macrophages. So if they're inside the macrophage, they won't be eaten by neutrophils. Most of the time, that protect them from destruction by neutrophils. And protect them from reactive oxygen species in general. Another thing is that for some bacteria, Listeria, for example, has been shown to actually change the type of cytokines macrophages will produce. So they will alter the immune response to favor them. Instead of a Th1 response, they will skew it towards something that favors their survival, okay? So again, changing the cytokines so the immune response is better for their survival, okay? And the third one is the horse. It's a dissemination. Once you are hiding within the macrophage or the immune cells, you can go wherever you want. None other immune cell will find you, theoretically. So it helps basically the theory of Trojan horse. You might go back to classical Greek literature if you don't know the story, but hiding within the... I will not tell you the story. You will look at it if you don't know it. But basically, the concept is hiding within the enemy, okay? And then hiding yourself from the enemy to better beat it. So when you're inside the macrophage, you can move within the tissue. Maybe you can go from one organ to the other through this pathway. And on the way, no one will ask you anything, right? You're hidden within the macrophage. No complement, no antibody will kill you. You're hidden, okay? So that's the Trojan horse theory of that. Using the cell to move you around, basically. All right. So that's it for this review of the immune response to bacteria, okay? Again, what's more important here is are the concepts. I don't need you to remember the different protein expressed by bacteria or things like that. Okay? You have anyone has a question? Or